morning, everyone. Welcome to today's lesson, which we are talking about cell division, which is super exciting. So we've been learning a lot about DNA replication and what the DNA is made out of, and we've touched briefly on our RNA as well. Today we're going to be talking about cell division, what happens during cell division, how that links to the DNA replication, and then also that is going to lead into what we're doing next, which is talking about genes and how we get genes from parents and stuff like that. So first thing we need to know is what is cell division, what is meiosis, and of course how do you say meiosis, which you hopefully should know since I just said it. Anyway, first slide, cellular replication. There are two main ways for cells to replicate. You should know this, you should have heard of it before. If not, the two ways are mitosis and meiosis. And we will be going through both of these very shortly. We're going to briefly talk mitosis, but meiosis is the main focus for the video. So starting with mitosis. Mitosis occurs in all cells except for the germ cells, which are the sex cells, sperm and eggs. It occurs mainly to allow for growth and to replace cells that are worn. So your skin cells are doing mitosis constantly. They are constantly dividing and replicating and creating new skin cells so that the skin cells on the top layer are able to be shed, I guess. They're able to die off and we lose them and stuff like that. Also things like hair and toenails and fingernails, they are constantly replicating and duplicating so that they can grow. So that's what mitosis is for. It's pretty much every part of the body except for the sperm cells and the egg cells. And one of the ways that I remember that is mitosis. So it happens in my toes, sis. Good times. So essentially this entire process, the purpose of it is to get two genetically identical daughter cells. So you want all of your hair cells to genetically be the same. If the genetics of your hair cells were changing, then you'd have splotches of green and blue and brown and black and whatever in your hair. That is just natural differences. I hope that makes sense. So when you're growing things, you don't want it to change randomly as it goes. You want it to be exactly the same, so you have the same color hair all throughout your hair strand. I think that made sense. And then over here, you can see the cursor. This is two cells, and this is cells in various stages of separation. All right, so the stages of mitosis, we're not really going to focus on them very much, but there are 6 main phases. So the first phase is interphase, which is kind of the in-between phase where things will grow and develop and get to a regular size before it then splits into 2. If it didn't have that phase, you can kind of tell in this picture, but these cells here are a little bit smaller than these cells over here. So if these ones then replicated again, the next cells would be smaller and then smaller and smaller, and then eventually we'd have tiny cells, and that would be kind of pointless. So interphase is the phase that kind of is between cytokinesis and prophase that enables the daughter cells from the last separation to grow again to get to a point where they're a big enough size that they can then divide. Prophase is where all of the genetic material starts to kind of combine together so it can be pulled apart properly. Then there's late prophase where it kind of lines up, it gets attached to these things called spindles, which are the white things, which is what helps pull it apart. Metaphase, it lines up in the middle of the cell, and it doesn't exactly perfectly line up like this every time, but it will line up semi-close to the middle of the cell. Anaphase is when the different parts get pulled apart by these little spindly thingies. Telophase is where it kind of starts, it forms a little bit of a nuclear membrane around that genetic material that just got pulled apart, and it starts to split off. And then cytokinesis is when the two cells finally completely cut off each other and the genetic material goes back to how it is usually. Hopefully that all made sense. And that's all we're talking about for mitosis. You don't really need to know much about mitosis, and it is very similar to meiosis in the steps. So we're going to go through in more detail those steps that we just talked about and how that relates to meiosis, but it is very similar to the process of mitosis. Hopefully this is all making sense and you're all following along. So, meiosis. This is the process of cell division for the sperm and egg cells. This is very, very important. It creates 4 different cells and each of those different cells will have a slightly different combination of genetic material compared to the original cell. So in meiosis, you don't want your egg cells or sperm cells to be exact genetic replicas of yourself, because that's not promoting diversity within the species. That is making everyone would be identical, essentially. So what we kind of want is 4 cells that have a different combination of the mother's genes, and then from the father, cells that have different combinations from the father's and mother's genes, so that each of those cells, when it combines with another cell, will create a unique human being. I hope that makes sense. It's one of the reasons why you and your siblings probably don't look exactly alike unless you are twins. So each daughter cell will have 23 chromosomes in it. If you have forgotten, a chromosome is one of these things. We will talk a little bit more about that, I think. So we start here with a cell that's got 2 chromosomes in it. It splits, so it then has 2 cells, 1 with each in, and then that splits again, so it's got 1 of each. So in this stage, the original kind of blue and red chromosomes duplicated itself to create an exact copy, and then that gets pulled apart and pulled apart. I hope this is going to make more sense as we go through. So, main phases of meiosis. We start again with interphase. So, as I said before, it is important because it allows time for it to grow and then get prepared to get started. So, during this stage, the DNA is duplicated, so it is directly copied. So, if we look down here, this is a picture, I think from Khan Academy. We've got, this is chromosome 1. It is directly duplicated here to have a second version of chromosome 1. Same with chromosome 2, 3, and 4. Or sorry, chromatid. So they're all replicated down here, so you've got 2 exact copies of each one. And that produces the identical copies, which we call sister chromatids. And they kind of attach at this little thing here. This little purple dot here is called the centromere. So they're actually physically attached to each other at one point, and they create this kind of weird X shape. So that's interphase. The second phase is prophase. So during this phase, the sister chromatids line up with their homologous chromosomes. So each one of your cells will have 1 copy of a chromosome from your mother and 1 copy of a chromosome from your father. So in this case, the red is 1 copy, let's say, from the mother, and the blue is a copy from the father. So they've each duplicated to create this second sister chromatid, which is like the exact replica of it. So both of the red ones are an exact copy from your mother, and the blue ones are the exact copy from the father. Hope that makes sense. Then these are like the centromeres that are sticking things together. And then there's also these like little stringy things which are sticking the 2 homologous chromosomes together. That was so hard. So they line up with their chromosome partners, and there's a thing that happens in this phase which is very, very important, and this is one of the main reasons that we have a great amount of genetic diversity that we have. So it's a process called crossing over. So crossing over is a process where the genetic material from one chromosome trades with the genetic material from the other within the homologous pair. So we have our mothers. So when I'm talking about mothers and fathers, I'm talking about in your cell that is going through meiosis, your mothers gave you half of the genes and your father gave you half of the genes. So the blue chromosome originally came from your mother into you, and the red chromosome originally came from your father. So in this last section, I showed you these little white lines where they're kind of sticking together. During this process, the way that they stick together, when they get pulled apart, they can get pulled apart unevenly because certain sections can get swapped. So you can see down here, this little section at the bottom has done this crossing over process and it's swapped. So now the mother's chromosome that is in you has gotten a tiny little bit of the father's chromosome, and the father's chromosome has gotten a tiny little bit of the mother. I hope that makes sense. So what essentially I'm saying is when one of these chromosomes is going into the final egg cell or sperm cell, it's not going to be an exact copy from your mother or your father. It's going to be a combination of your mother and your father that is combined in a unique way. So the result is an exchange of genetic material, and it allows for genetic variation. This is very, very important. Genetic variation. The whole purpose of this is variation in the species. Now that's very important, because if there's no diversity and variation in our species, if something comes along and attacks a specific gene that is in one person and there's no variation, that specific gene is going to be in every person. So anybody with that gene is going to have the consequence of whatever that problem is. If you've got genetic diversity, that one person might have that gene that gets attacked, but a lot of other people might have a slightly different variation of that gene, or a completely different variation of the gene, so won't get the consequences of that disease. I hope that makes sense. So this crossing over process is very important to increase the variation and diversity in the species. So that's the second phase, which is prophase. The third phase is called metaphase. So during this phase, they, like I said before, line up in the center of the cell. We have some of these crossing overs happening. They haven't pulled apart yet, so it isn't combined into a kind of full, it hasn't completely separated yet, but you can see that it's kind of gotten to the point where when they separate, you can see where it's going to separate. So they line up in the center, the little spindles attach, the centromeres down here are sending out the little spindles. Some of them, like you can see over here, don't have the chromosomes attached to them. They are instead kind of attached to each other. So the way this works is the ones that are attached to each other kind of push onto each other, hold the centromeres away from each other, so that when these ones pull back, it pulls the chromosomes away from each other without pulling these closer to each other. So it uses these to kind of center itself in the cell so it stays put. I hope that makes sense. Anyway, getting too far ahead of ourselves. So metaphase is the phase where they line up in a row. It can be completely random which side it is. So the mother's genes aren't always on the top and the father's genes are always on the bottom. No, sometimes the mother's on top, sometimes the father's on top. Hope that makes sense. And that also increases the genetic diversity of the species. Cool. I think this is kind of semi-making sense. Next phase, anaphase. The chromosome pairs are pulled apart to opposite sides of the cell. So the chromatids remain tightly bound together at the centromere, but the chromosomes pull apart. So you can see here, the chromatids are still attached, which is just this little, I don't know, balloon animal looking thing. And it's got the little bits that have done the crossover process are now fully separated from their original chromatid and are now on their brand new chromatid. I'm hoping this makes sense. And it wouldn't necessarily just be one thing or two things. It might be quite a lot of things that have swapped over, but there will definitely be some swapping over happening. Hope that makes sense. All right, telophase. This is where the chromosomes, they were in the center, they've been pulled apart. This is a real picture, by the way. And they kind of move towards the center of where their new cell will eventually be. This allows for the next stage of separation to happen. So if this was mitosis, and I do think I've got the next slide is about, yeah. So if this was mitosis, this would be kind of the section where this entire nuclear envelope will reform, and all of the organelles that have also been pulled apart, if there were any, they will kind of separate off to where they're meant to be. In the case of meiosis, sometimes this doesn't happen fully, sometimes it does, but it is the physical separation of the components into two daughter cells. So at the moment, each one of these daughter cells, which is what we call an individual cell that's come from this, they're not exactly identical, but they have the same amount of chromosomes as the original cell had. So the original cell would have 23 pairs of chromosomes. Each one of these cells will still have 23 pairs. The next stage is to go from having 23 pairs to having half of that, so just 23 single chromosomes. Because if you had 23 pairs that came from your mother and 23 pairs that came from your father, you would have 46 and would all of a sudden be a brand new species. Don't want that. So we need half of this so that when reproduction is occurring, you have half of the cells or genetic material from your mother, half from your father, and that gives a full human being. Hope that makes sense. All right, meiosis 1. That is the first stage of meiosis. So that's pretty similar to mitosis. The only thing that's not the same with this as mitosis is that none of the crossing over will occur in mitosis because you'll get exactly identical pairs. In this, you don't have that process. So the crossing over gives you slightly different combinations of genetic material, but you end up with two cells that have the same number of genetic material. Right, so this is kind of, I'm pretty sure this is definitely from Khan Academy. This is a diagram that shows those stages, and it shows prophase 1, metaphase 1, anaphase 1, and telophase 1. It would probably be a good idea to draw this out yourself, either as just a diagram or make it a full poster, and kind of get used to the idea of exactly what's happening within that cell. It's kind of important to understand. So give yourself a go, do that, make a poster, make some make some flashcards, or just draw it out in your book. Okay, now we get on to stage 2. So we go through the exact same steps again. So we're going to start with prophase 2. So if it has completely split apart like this and they've decondensed into just like strings, which actually kind of like this, they will recondense again into their little chromosome pairs. The nuclear envelope, if it's formed, it will break up again, and the centromeres will move to this new section on opposite sides of the cell. Metaphase 2, the chromosomes will line up in the center of the cell again. This time we don't have duplicates, so we've only got the individual chromosomes, not pairs of chromosomes. So this time when they pull apart, we'll be pulling apart the sister chromatids, not the pairs of chromosomes. That's very important. So metaphase 2, they line up in the center and the spindles are formed and attached to the chromosomes. And again, we've got these little ones here that are attaching to each other to kind of give resistance so they don't get pulled too close to each other. Anaphase 2, the sister chromatids are pulled apart. So they are pulled apart from the centromere and become individual little balloon animal balloons. They've got a single sister chromatid that is getting pulled to each side of the cell. Telophase and cytophase kinesis 2. No, telophase and cytokinesis 2. So we've pulled apart the sister chromatids, we pull them into their separate little sections, and then we chop off the cells, or cleave apart the cells, I think is the term, and it becomes 4 different cells. Each of these sister chromatids will unravel and become a bowl of spaghetti looking thing, which is just the DNA all floating around in the nucleus. It doesn't sit in these condensed forms. It just, I don't know, unravels like this. And it looks like a huge giant paint splatter of a mess, but all of the DNA and genetic material is contained in there. And that's in there, that's there. This is the important part, each of the 4 daughter cells contains half of the amount of chromosomes. So, as I said before, each human being has 23 pairs or 46 chromosomes, and then each egg or sperm, which is one of these ones, only has 23 individual chromosomes. It does not have pairs. Hope that makes sense. So in each one of these cells would be 23, so we've got 23, 23, 23, and 23, and then each one of the original cells would be 46 and 46. So 46 splits up into 23. And then again, this is the rest of the diagram that has the prophase, metaphase, anaphase, and telophase 2. So same thing, recondenses here, lines up, pulls itself apart, and then we've got the different cells. And you can see in this diagram that each one is genetically slightly different, has a slightly different combination of, what is this called? Pink and purple colors, which represent where they came from originally. And you'll notice here, this little small tiny one, that would be a Y chromosome. So this would be a male, and this also represents kind of the male section. So hopefully that kind of makes sense. I think it might have. So this is the full overview. Your job now is to create your own full overview of this process. So it can be a poster, it can be your own PowerPoint, it can be flashcards, it can just be a diagram with color coding in your book, whatever you want to do. But it doesn't need to be an exact copy of this, but you've got to try and create your own full overview of the process of meiosis, because that's where you're going to find out what you're struggling to understand, and then you'll be able to work out how can I fix my understanding of this. If you go through this and you understand it all and you can make your own little summary sentences underneath and all of that, and you can draw your pictures fine, that's when you know that you're at the stage that you understand it. I hope that makes sense. I think that's the last slide. So I hope that you got something out of this video. Sorry if it was a bit confusing. You are welcome to ask any questions that you want, and I will attempt to answer them to the best of my ability. Anyway, I shall see you later. Bye.